Hey guys, welcome back. This is episode 3 of the topic 1, DNA and proteins. And today in 1.3, we're going to be covering protein structure and function. Now, this is a really important concept that comes back in topic 2, it comes back in topic 3, and it comes back in topic 4. And I guarantee it'll be tested in your exam. So make sure you know this inside out. So biological molecules, they all have structures or shapes, and they all have functions. And these two concepts are interconnected, and you need to understand that. So examples of proteins with specific shapes include enzymes, hormones, receptor proteins, and antibodies. These biological molecules' functions are dictated by the shape of their functional site, which is known in biology as an active site. In chemistry, it's called a functional group. In biology, it's called an active site. And in stage 2 bio, the relationship between the structure and function is typically taught using the example of enzymes, which I'm sure you've heard of a lot of times before. So enzymes are biological catalysts, and they're specific and complementary in shape to their target molecules known as substrates. So the diagram shown here is a really common diagram, drawing these enzymes as circles and the substrate as this other pointy shape there. You'll see this a lot of times, and you'll kind of understand it the more you see it. Enzymes, as catalysts, increase the rate of chemical reactions by lowering the activation energy, which is the energy required for a reaction to proceed. Now, if you're doing stage 2 chem, you need to be able to understand that in chemistry, you can't say that enzymes lower activation energy. You say that they offer an alternative pathway which has a lower activation energy. And that's just the nature of how chemistry explains collision theory and rates of reaction. But in biology, the subject outline explicitly states enzymes lower activation energy. So the mechanism of an enzyme-catalyzed reaction can be described using the induced fit model, which is something that we see quite a lot in stage 2 bio throughout all 4 of the topics. So the induced fit model basically involves an enzyme and a substrate binding together to facilitate a reaction. It's a 4-step process. I would recommend, again, learning the 4 steps. So any time that I create a numbered list of steps, it means I want you to learn the steps that are involved because they're going to be tested, they're going to be examinable, and they're really high yield concepts. So, firstly, the substrate binds to the active site of its specific or complementary enzyme. And you can see what complementary means in the diagram here. They kind of fit together like a lock and key. The active site induces a complementary fit of even higher specificity. So you can see in the diagram on the left, the enzyme and substrate are kind of complementary, but on the right, they're completely 100% complementary. That's what this induced fit is. So the induced fit places a strain on the substrate, weakening the interactions that hold it together. So the substrate is held together by a bunch of different chemical bonds, but these are all weakened by the pressure and the stress that's exerted by the induced fit. So because these molecules are now a lot weaker, less energy is required to break it down. So I'm talking about the substrate here, right? So this essentially reduces the activation energy of breaking down the substrate, which allows the reaction to occur at a much faster rate. So this is a really simple but yet really effective diagram that describes the induced fit model. You'll see it time and time again, and I think every year for the first prac, which is the enzyme prac, every student includes this diagram. So just a little tip here, don't include it in your prac because it's cliche, it's overused, and teachers hate it. And if you want to be hated by your teachers, trust me, I've had so much experience with that, do not include things that are cliched and overdone. You can make your own diagram or you can use something similar, but yeah, don't include this one because everyone's seen it. So there are a lot of factors that affect the rate of enzyme-catalyzed reactions, and we're going to cover a few here. So the first one is temperature. The rate of reaction initially increases with temperature as both the enzyme and the substrate gain kinetic energy. So this effectively increases the number of collisions between the substrate and the active site of the enzyme. The rate of reaction is greatest at the optimum temperature, Now you can see it's shown in green on the diagram here. It's basically the peak of your bell curve. It's the local maximum, as you would call it in maths. The rate of reaction decreases as we go past the optimum, as the attractive forces holding together the tertiary structure, so remember the 3D structure of the enzyme, are overcome, and the enzyme is denatured, which means it loses its biological shape and its biological function. The substrate and enzyme concentration also has a big impact on the rate of reaction. So the rate of reaction is really low when enzyme and substrate concentration is low, as less particles are present to potentially collide. The rate of reaction increases with the enzyme and substrate concentration as more active sites and or substrates become available to form these enzyme-substrate complexes. The rate of reaction becomes constant at point X, which is shown on the diagram, as either the enzyme or the substrate become the limiting factor. So another factor that we look at is pH. So all enzymes are located in different parts of the body of a particular organism. Now what that means is that they function best in particular environments, and pH is no exception. So enzymes found in the stomach typically function best in acidic conditions because the stomach is naturally acidic, so they need to be able to acclimate to that environment. So what this means is that enzymes in the stomach perform well at low pH, because remember, low pH equals more acidic, high pH equals more basic conditions. And this means that the pH optimum of different enzymes is completely different depending on the enzyme. You don't need to memorize any, but you need to be able to explain why it is that certain enzymes perform better in certain environments. So another thing that we talk about as a factor affecting the rate of enzyme reactions is chemical inhibitors. So the activity of enzymes is tightly regulated in cells by molecules known as inhibitors that inactivate enzymes. So the term inhibitor basically means any substance that interferes with enzyme activity. But in stage 2, we look at two types of inhibitor: competitive and non-competitive, or allosteric inhibitors. Both species of inhibitor affect the reaction graphs as shown on the right. So obviously, competitive inhibitors reduce the reaction rate a little bit, and non-competitive inhibitors completely reduce the reaction rate quite a lot. You don't need to know how to draw this graph, but it's just something I thought I'd include, just to help you kind of understand how inhibitors work. So competitive inhibitors are structurally similar to the substrate. In other words, they're complementary to the active site. And because of this, they compete with the active site with the substrate. They form what's known as an enzyme inhibitor complex that prevents the formation of enzyme substrate complexes. So because of this, competitive inhibitors inhibit enzyme action and therefore decrease the rate of the enzyme catalyzed reactions. This diagram here explains it pretty well, and you kind of just need to know that it occupies the active site because of a similar shape to the substrate. Non-competitive inhibitors, on the other hand, bind and attach to enzymes at a site called the allosteric site. An allosteric site is basically anywhere on the surface of an enzyme excluding its active site. So basically anywhere that's not the functional active site of the enzyme. Binding of the inhibitor to the allosteric site causes the enzyme's active site to change shape. So again, it denatures, and this inhibits the formation of enzyme substrate complexes. Non-competitive inhibitors therefore also inhibit enzyme action, thus decreasing the rate of enzyme catalyzed reactions. Okay, we've covered all of the factors that affect enzyme reactions, and that's basically all there is for structure and function. Remember, this doesn't only apply to enzymes, but it also reacts to other biological molecules like proteins, antibodies, and hormones. So if you ever get a question about the denaturing of a protein or the changing in the shape of an antibody, just remember we can apply these same principles that we've learned with enzymes to those molecules, because biological structure and function are interconnected and it's universal for all protein molecules. All right, thanks guys. See you next time.